Good morning, good afternoon, everyone, and welcome to today's Exploring by the Seat of Your Pants event. My name is Joe Grabowski, and I'll be your host for today. I know many groups are joining us in multiple events this week, and you've heard this a lot. It is one crazy week here at Exploring by the Seat of Your Pants. The school year is coming to an end, so we've lost some classrooms in the U.S., and throughout the month of June, leading up right to the end, classrooms across the U.S. and Canada will start to go out on their summer vacations. But this week, we are throwing a lot of content your way. It is World Ocean Week. It's Canadian Environment Week. And we're also featuring early career ocean professionals as well. So it is a loaded week with 25 or so live events. I think it's actually pushing 30. But we're really excited to have this opportunity to share with your classrooms. And so we are celebrating Canadian Environment Week. This is the first time we've ever done this festival before. It's through some funding from Environment and Climate Change Canada, who are an amazing partner to work with. And we are really looking at the environments that make Canada, Canada. So looking at the air, looking at the forests, the Arctic, the freshwater, and the ocean ecosystems. We have a ton of great content, a lot of great researchers and filmmakers and scientists joining us from coast to coast to coast. So if you would like to know a little bit more, if you'd like to join in throughout this week, if you visit our website, /Environment Week, you will find all the events we have coming up, and the links that we share can be used to watch the recordings of the events as well. So this event right now is going to focus on our freshwater ecosystems, and we have Neil Mochnacz returning to join us. He joined us a few months ago to share some of his work. He is a research scientist with Fisheries and Oceans Canada in Winnipeg, Manitoba. He joined the department in 2003, and his research really looks at understanding physiology, landscape, climate, and biophysical factors, how they affect populations of river fish in central and northern Canada. So his research is looking at improving our understanding of delivering science through things like support, monitoring, recovery, threat assessment of threatened and endangered freshwater fish species. So let's bring Neil in live with us right now. Hey, Neil, how you doing today?
Good, thanks. Thanks for having me.
Of course, Neil, it's great to see you again. It's great to have you representing freshwater ecosystems during our Canadian Environment Week. We've got a great group of classrooms, so we'll take questions in the chat today. We'll play Kahoot today. It's going to be busy. So Neil, I'll let you take over for a little bit. And yeah, I know I've got a couple of videos to play for you, so just give me a shout when you want the next video and we'll get right into it.
Great. Thank you very much. Just get things going here, and thanks for having me. It's a real pleasure to be able to be here for this week. So everybody see my screen okay?
I see it loading up backstage. I'm just going to wait till it goes full screen. There we go. We're good.
Perfect. Great. Yeah, so today I'm going to talk about the work I do and importance of conserving Canada's freshwater fish. So I'm a research scientist with the department. I also have a family. That's my wife, Carly, and I have 3 kids. Abby is the oldest in the middle. Dax is 16 and Avery's on the left. And then we have our little dog, Willow, which is part of our family. So I just wanted to talk a little bit about the work that I do and some of the different types of fish that I do work on and where I work. So this is a map of North America. And I work sort of in 3 areas of Canada. I work in Alberta, which is the yellow dot you see there. I also work in the Northwest Territories. So those are the blue dots in the upper part of the map. And I get to work on some pretty cool fish. So I work on species of fish called trout, char, and grayling. So trout, one of the types of species I work on is the one in the top. So those are sort of the red and orange fish. That species is called Dolly Varden. The fish that I'm holding in the picture below is called a bull trout. It's quite closely related to Dolly Varden. And then the fish in the lower right-hand corner is called an Arctic grayling, and they're a really cool fish. They have this really neat dorsal fin that looks kind of like a sail. And when you bring them out of the water, they're iridescent. So they have these different colors that reflect off their bodies, and they're pretty silvery. So yeah, I'm very fortunate to be able to work on some really cool fish. They look cool, and they're very important to Canadians, and they occur in fresh water. The other really cool part of my job, though probably the coolest part of my job, is I get to go to a lot of cool places and see a lot of cool things. I work mostly in rivers, and I get to see cool things like these waterfalls. This is another cool spot that we actually landed a helicopter on the top of this rock in the middle of the screen here, and we were doing some work to put some receivers in this river so we could track where fish go. And then this is another really cool vista or shot. So flying helicopters, even though they're just the way we get to places, I'm very fortunate. I get to see some really cool places and some cool sites. Like I said, we primarily use helicopters because a lot of the areas we go to are really rugged, mountainous regions, and so we need a helicopter to drop us off where we could do our work in these areas. So why is it important to conserve Canada's freshwater fish? Well, I don't know about you guys, but in most instances, anytime I'm fishing or I'm fishing with people, generally, even if you don't like fishing, when we catch big fish, everyone just loves catching big fish. It typically is fun to do, but also makes people really happy. And I think this is a great example of it. This was on one of the projects that I was leading, and we were putting tags into these fish and then tracking where they go. And this was one of the biggest bull trout that we caught for the program. And as you can see, everybody's pretty happy. And that's what I think is the coolest part of my job is to get to work on these fish and make sure that they're around for future generations. This is another project that I worked on. This is in Alberta. And what we were doing here is we were capturing these fish when they're getting ready to spawn. So they're getting ready to lay their eggs. So this is a bull trout population. And in the picture in the right-hand side, those cleared areas you can hopefully make out in the picture, those are areas where the fish have excavated what we call redds. So what happens with this particular species is when they're getting ready to lay their eggs in the river, the female moves a bunch of rocks out of the way and gets ready to lay her eggs, and then the male comes and partners up with her, and they do their thing. They lay their eggs. The female covers the eggs back up with rocks. They remain in the rocks for from the fall all the way through to the spring, and then in the spring the eggs hatch out and the juveniles emerge, and we have baby fish to contribute to the population. So the species that I work on are a class of cold water fishes. So they require cold water in order to persist. And that's very important for fish because fish are called aquatic ectotherms. And what that means is they regulate their body temperature using external sources. So whatever they're in, whatever medium they're in, or whether they're, in this instance, water, the water's cold, that's typically the fish are cold. Whereas people like us, like mammals, we're able to regulate our body temperature based on our internal organs and our physiology. Fish are their body temperature and how they operate from a physiological perspective, how they generate energy, how they generate warmth. That's based on where they are in their environment, in this instance, rivers. Why is that important? Well, for ectotherms and fish, it's very important that we understand the range of temperatures that they require because they are ectotherms, because our world is heating up, right? Everybody that's been paying attention and knows a little bit, I'm sure you guys have talked about climate warming, climate change. We continually see our world warming up, and it's going to continue to do that. So understanding how that may affect species like the ones I study that require cold water to persist is really important. And it's important because, as I mentioned, different types of fish, the fish that I do work on, they're called cold water fishes. We want to understand the range of temperatures in the rivers that they occupy, what those range of temperatures are, and how that affects things like growth. And as temperatures change and maybe go beyond the optimal range of temperatures that these fish require, we need to understand that so we can see how that affects things like growth. So one of the other things that we do is we look at where we find fish across the landscape, the aquatic landscape or rivers. But we also want to understand which type of areas or habitats fish use. And so what we do is we look at movement patterns of fish throughout river systems. And so what we do is we put tags in fish that send off a signal, and then we put out receivers. And anytime the fish goes near one of those receivers, it records which fish was there, when they were there, and for how long they're there. And then we can better understand the different areas across a river system, like you see in the picture here, that fish use. We also look at habitat use and what fish are doing when they're in particular areas. So when we see fish across the aquatic landscape, we want to understand, are they moving between feeding areas? Are they searching for food? Are they moving to areas where they're going to lay their eggs? Are they moving to different areas where they can feed, or are they in areas where we see juveniles growing up and residing so that they can be part of the adult population at some point in time? So another question that people often ask me is, how do you catch fish? And one of the main ways that we catch fish is using backpack electrofishers. So I'm in the picture in the middle, and I have this Ghostbusters type of backpack on. And what this backpack does is it puts an electrical current through the water. It doesn't hurt the fish, but what it does is it temporarily stuns the fish. And when the fish gets stunned, they flip upside down for just a second. We scoop them up with a net. We put them into a bucket of clean, fresh water, make sure they're okay, and they typically always are. And then we can take measurements. We can identify which fish they are, and then we can send them on their way. And so, Joe, if you could show the first video, that would be excellent.
Yeah, we'll control through rest, but then do
Hello, bonjour, merci, welcome to Nahanni National Park Preserve. My name's Sarah, I'm an ecologist for the park.
My name's AJ, I'm a technician working for Fisheries and Oceans Canada.
So we're out here on the very upper end of Wrigley Creek looking for bull trout.
Bull trout are the fish we all care about. Important cold water species that's an important indicator for lots of other fish species in the park.
We use them to help us monitor what's going on in the park, if there's any changes happening to our ecosystems.
We're using several different methods to catch fish on this project. Electrofishing is one of them. So I got the magic wand here and my Ghostbusters backpack.
And then I'm gonna be following behind AJ, and I'm kind of like the goalie. Gotta catch those fish as they come downstream towards me.
There we go. So we've captured our first bull trout for the day.
Woohoo!
Yeah. So we got a nice size one here. 22.
The other technique that we're using is something a little bit more recently developed called environmental DNA, or eDNA. We don't need to see or catch the fish to know that they're there. We can just take a water sample and then ship them off to the Fisheries and Oceans Genetics Lab in Winnipeg. And they process the samples and tell us if there were bull trout in this stream or not.
One of the best parts of this job is just getting to sit in the pools to cool down on a hot day of sampling.
Absolutely, and I reckon we deserve it. It's been like a busy, very full-on week, but it's been a great time. So it's the end of the day, like there's the helicopter coming in to pick us up. Another successful day, small bull trout.
One of the things people always ask me up north is what's the light like up at night? Well, here we are looking out at the sunset, and it's 12:30 in the morning.
So it's our last field morning today. Myself and one DFO will be sticking around camp to do some sample processing. Let's go get the day started.
Sarah, come check it out. Our results are in. Check it out. So,
What am I looking at?
looks like the reds, our controls were working. Didn't see much with the greens. Nothing in the next 3 samples. Whew, bull trout.
Really cool to see that happening just like in the field while we're sitting in this cabin. Like,
Absolutely.
not having to wait, you know, as long.
Great. I hope you guys enjoyed that video. I'm going to move on to talk a little bit more about some of the things we do, and then I'll show one last video. Yeah, so I think that was hopefully you got a better appreciation for how we catch the fish there. As I mentioned earlier, we also, a big component of what we do is tagging fish and then tracking where we find them. And so we capture fish, and then we typically surgically implant an acoustic tag inside the fish. We sew them back up, and it doesn't hurt the fish, and they're fine. And then we put out these things, so these black receivers that you see in the middle, attach them to rocks or cinder blocks, and that we anchor them to the bottom. And then every time a fish moves past one of these things, we can detect that fish and determine where they're moving. So this is just an example, a little animation of what we would see for one fish after we've tagged the fish, and you would track them over time. And so it's a really powerful tool that we can use to understand where fish go, the different habitats they use during different times of the year. So this fish went all over the place in sort of the one area of the lake, which is pretty cool to see, and then made some sort of longer, bigger movements down over into sort of the right-hand part of the arm of that lake. So the other really important part of what I do is that the species that I study, like bull trout, are called sentinel species. So, or another way to put it is they're indicator species. So if the aquatic ecosystem that they occur in, or the rivers they occur in, are healthy, are not polluted or in good shape, then typically the fish will be in good shape. So they're an indication of the broader ecosystem health or the health of the river systems that they live in. And so if we can monitor bull trout, these indicator species, we can work together with people to understand the status of these populations, but also the health of the broader river system or ecosystem. And so that's really important, because that's a really powerful tool we can use as a early warning system for anything that might be negatively affecting these systems. So Joe, if you could show the last video, that would be great.
Sorry, Neil, just give me a second. For some reason, I'm getting an error message when I try and hit play.
Okay.
Maybe I'll go right to YouTube and I'll play it from there. Just bear with me for 1 second.
Yeah, no problem.
And I will get it up for us. Okay. Here we go. I'll share my screen. Do a Chrome tab. Perfect. Just let me know if you can hear okay, Neil, but I think it'll probably be okay.
Yeah, it looks good. I can hear it.
Perfect.
Hey everyone, Michael Short here. Come on, let's go outdoors. Well, welcome to the Kananaskis area, where an interesting study is taking place looking at bull trout and whether or not bull trout eggs can be transplanted into streams like this. Of course, there have been many challenges facing bull trout here in Alberta.
So bull trout are Alberta's provincial fish, and they're kind of like the grizzly bears of our mountain and foothill rivers. They're often the top predator on the food chain. They can get to very large sizes, and they have silvery missile-shaped bodies with pale spots on the side. In their spawning season, they develop bright white leading edges on their fins, and the males grow big hooked lower jaws called a kype, very similar to a salmon that you'd see on the coast. Unfortunately, many populations of bull trout across the province aren't doing very well right now. And depending on where you are, it could be a combination of one or many things. In some cases, it's habitat degradation. In others, it's overfishing, and maybe even competition and hybridization with stocked non-native eastern brook trout. So really, the right recipe for recovery really depends on what population that we're hoping to recover. Recovery stocking is different than recreational stocking that most people are familiar with. Recreational stocking is done to create a fishery for people to enjoy, whereas recovery stocking is done to conserve a species and in some cases even bring it back from the brink of extinction. Here in Alberta, we typically do recovery stocking to address a couple key threats: climate change and competition and hybridization with stocked eastern brook trout.
Partnerships play a crucial role in the success of a bull trout recovery program. Over the next few days, fishery biologists from EPA will be collaborating with counterparts from DFO, the University of Calgary, the Alberta Conservation Association, and FRI Research, which are one of the collaborators for Native Trout Recovery Program.
Partnerships and volunteers are absolutely critical to native trout recovery. The problems that we need to address to recover native trout are far too complex for us to do by ourselves. And there are many different ways that people can get involved in improving the lives of native trout. And it could be as simple as, if you're an angler, make sure you're handling those fish properly. Or if you want to get your hands dirty, check out our partner websites, Trout Unlimited Canada or Cows and Fish, and come out for a day and help plant willows and make habitat even better.
Bull trout swim up this creek to spawn, which allows biologists to use electrofishing to catch enough males and females. Once caught, the fish are held in a netted off area in a deep pool. So the idea here is that folks are wearing electropack, going along the banks and just bringing out these monster bull trout. I've never seen anything like it, and I've been fishing for, I don't know, an awful lot of years here in Alberta, but this is simply amazing to see. One of the contributing factors to the growth of this population of bull trout is the angling restrictions introduced in the 90s. Since then, the government of Alberta and organizations like the Alberta Conservation Association have been keeping tabs on this critical spawning creek.
Yeah, so ACA has some history out here. So we supported a project on the population estimates work done in the Upper Kananaskis bull trout population, late 90s, early 2000s. So with the closure out here, it's really just nice to see the amount and the healthy size of the fish that we were seeing the last couple of days.
After collecting a number of bull trout, the next step is to harvest the eggs from the females and the milt from the males. However, biologists need to ensure that the females are ready to lay their eggs before this process can take place.
So I've never seen beautiful eggs.
Yes.
Look at the size of it.
See that?
Wow. All the way back. Yep.
That's in line.
And just how many eggs will a single female bull trout lay?
Well, you really put me on the spot here, Michael, but I would estimate around 2,400 eggs per female on the higher side.
And when it comes to the milt needed to fertilize the eggs, one has to ask, is that the volume or the potency that biologists are looking for?
That's a tremendous question. Visually, you can ascertain on the milt sort of the potency. If it's a really solid white, you might put in less. Other stuff is pretty watery, so we were able to use probably about a cc from each male for the fertilization. Too much milt is no good. It'll actually choke the microfile and allow for a poorer fertilization. And if you don't have enough, there's just not enough sperm to actually swim to each egg. So it's kind of a fine line, more of a learn as you go and from just historical knowledge.
In order to ensure genetic diversity, the milt from 4 male bull trout is being used to fertilize these eggs.
And if we did have one male with poor milt, we can ensure fertilization to the other 3. A little more, Lindsay, a little more, a little more, a little more, a little more. Keep going, keep going, keep going. A little faster, a little faster. There we go.
One other question that remains to be answered from this study is whether or not biologists can successfully plant bull trout eggs into suitable spawning locations.
So we're taking these eggs and going to put them in little capsules and be burying those capsules back in the stream to then see the survival rates over winter. So we'll pick them back up in the spring and see how many fish have survived. And this should give us a good idea of if this can be an effective method for conservation translocations. So obviously translocations aren't a universal solution, but we can use those in locations where the population's already extirpated.
So the first step today is a fisheries biologist is going to dig a hole about 20 or 30 centimeters deep, and that mimics exactly what that big female bull trout is going to do. Next step is we measure and make sure that we've achieved that right depth, and then we carefully drop the egg capsule into stream, and it looks a little bit like a hockey puck. Inside that egg capsule, there's 50 fertilized bull trout eggs and some gravel just to protect the eggs and make sure they stay well oxygenated throughout the winter time. Afterwards, a biologist will just gently cover that egg capsule with gravels once more. And again, this is exactly what would happen in nature with one of those big bull trout. So in 5 to 6 months, we'll be returning to site, digging up those egg capsules and seeing just how many eggs survive throughout the wintertime. And that's going to be the critical learning that we need to go forward and use this technique in other places across the East Slopes.
Biologists must learn how to identify the best spots in a stream for planting bull trout eggs. These fish can detect areas where groundwater seeps in, providing ideal conditions for incubating eggs. But factors such as substrate size, water depth, and temperature may all be crucial environmental conditions that must be evaluated. For this study, over 100 of these pucks will be planted into the stream bed at various locations. Department of Fisheries and Oceans, one of the key partners in this Kananaskis bull trout study, and they are here particularly to take some eggs back to Winnipeg, where they're going to look at what impact global warming may have on future production of bull trout.
And what we're going to be doing is we're going to be rearing those eggs under different temperature regimes. So we're going to fluctuate the temperature to simulate climate warming scenarios. As these streams warm, these eggs will be affected differently than they currently are. And so we're trying to understand if these streams warm by 4 degrees or 6 degrees during spawning time, how will that affect the viability, the survival, and development of the eggs? And that will help us understand what constitutes critical habitat, but also how to protect those habitats. I am very optimistic about the future for bull trout, mostly because of collaborations like this. We have so many different groups that are so passionate about conserving and protecting populations like these.
We should have an idea soon on whether or not these in-stream incubators will be successful. If they are, we'll have another tool to use in an effort to ensure bull trout will continue to be integral to the intricate balance of Alberta's ecosystem. Till next time, everyone, I'm Michael Short. Come on, let's go outdoors.
Great. I hope you enjoyed that that video and learn a little bit more about some of the things we do. I'm just going to share my screen. Joe, I think I'm mostly done here, so we can do questions. But yeah, I hope everybody enjoyed the videos and a little bit of some of the things that I presented. And I guess with that, I'll be happy to take questions.
All right, Neil, thanks for that great presentation and really highlighting some of the really cool work that you get to do out in the field. And just, some of that research is pretty hands-on, pretty in-depth, and a lot of information you're trying to capture. So it's really neat to be able to see the videos of that research in action. So what we're going to do before we start taking questions is we're going to play some Kahoot together. So I'm going to share my screen this time. I'm going to go to, let's see, there's the one that I want. You need to go to kahoot.it today if you're going to join us with your classroom, 691501, and we will play a little bit of Kahoot. I think I have 5 questions in there today. There's a mixture of multiple choice and true and false. I put 30 seconds in for each question, so you can think about your answer for a second. But the quicker you do get that answer in, the more points you are going to get. So the quicker you get it in, and if it's right, the more points we got coming your way. Well, we do wait to see if a few more classrooms are going to join us. I have a question here in the chat, Neil, and this is about those packs that you guys were using. I have a 3rd grade student who wants to know if they're heavy or hard to wear.
Yeah, they're not real difficult to wear. They're not as comfortable as like a backpack you would wear to school or that kind of thing. They are relatively heavy. They used to be a lot heavier because we have lithium-ion batteries now, so a different type of battery, the newest batteries, and they're a little lighter. But they're probably about 40 pounds, so 40 to 45 pounds.
All right. Well, let's get things started. We've got some classrooms in here ready to go. with their auto-generated animal names. So let's start our Kahoot. I think I did 5 questions this time. That feels like what I did. So 3-second count in. 3, 2, 1. And our first question is going to be a multiple choice. Which fish did Neil not mention that he works with? So we have trout, char, grayling, and bass. Which one did he not say that he works with? So let's get those answers locked in. Which one do you think it was? You've got a few more seconds on the clock. Remember, the quicker you get the right answer in, the more points you're going to get. I see we went mostly with bass. That's right. Absolutely correct. Let's slide over to our leaderboard. That puts the fast bison into the lead there. Neil uses helicopters to get to some really remote areas. Is that true? Or is that false, that Neil uses helicopters to get to some really remote areas that he works at in the field? 20 seconds to get an answer locked in. Get it in nice and fast, and you will be ready to go. All right. Good job, crew. That is absolutely true. Neil, some of those areas you get to work in are absolutely spectacular. And that's not too bad of a commuting vehicle either.
Yeah, I agree.
So that puts the awesome dolphin in first place. What's coming next? Spawning is when fish feed, rest, fight, or lay their eggs. Feed, rest, fight, or lay their eggs. So what are fish doing when they're spawning? I don't think it's like a video game when you respawn back into a video game. What do you think fish are doing when they're spawning? All right, that is correct. They are laying their eggs. What does that do for us? That puts the fast bison back in first place. We have a true and false. Fish can regulate their body temperatures just like mammals. Is that true? Or is that false, that fish can regulate their body temperature just like mammals? Got about halfway there to get that answer locked in. Can they regulate their body temperature? Are they warm or cold-blooded? All right, good job, crew. That's absolutely false. They cannot regulate their body temperature like mammals. We got the amusing buffalo moving into first place, and we have one more question to go. True or false, warming climate can impact the survival of cold water fish? All right, let's get that last answer locked in. If you are tuning in via YouTube, let's start putting some questions into the chat, and we will be ready to answer those questions for you today. A couple more seconds. There we go. That is true. Warming climate can impact the survival of cold water fish. They do need that cooler water. So in third place, we've got the amusing buffalo. In second place, the legend camel. And holding down that top spot, the freshwater. Oh, well, that's the fast bison. Sorry, I saw the title and thought it was a freshwater fish. It is the fast bison who has taken the top spot. I thought we got really lucky and had a fish name win today. Okay, thank you for playing Kahoot with us. I'm going to shut down that Kahoot and turn off that music. And I am going to start looking for some questions. So Neil, let's get right into it. This is from an 8th grade class in Toronto. They want to know, how long can eDNA last in water? So how long could the fish have been there and still you would know?
That's an excellent question. It's not a question that we really have a definitive answer for, because there's a lot of different things that affect how quickly it degrades or how quickly it's lost from the water and is no longer detectable. eDNA is typically like when a fish sheds cells off, it has like that mucus, that kind of thing. And depending where a fish is, it can last longer or less long. So in rivers, they're flowing water. So, the faster the water flows and the more water we have, that will affect how long eDNA stays in that river. And so it's a hard question to really answer, because it just depends on the type of environment that we see the fish in and that'll dictate how long it resides there and remains there. It's also the other element that's really important is how many individuals are there. Obviously, when there are more fish in a particular area, there's going to be a stronger signal and greater amount of eDNA in the area.
Okay.
Great question.
Yeah, absolutely. I've got a second grade class in Hamilton, and they would like to know about the wildlife in remote areas. Do you see things like bears?
Yes, another excellent question. We see all sorts of things when we're working in those remote areas. That's the different wildlife that live there. That's their home, and we do our best to try to not disturb those different animals. But we see things like moose, caribou, grizzly bears, black bears, you know, all different types of birds, falcons, eagles, that kind of thing. So yeah, it's pretty cool. It's really neat to see those different animals in their own environment or in their own home. So yeah, it's pretty cool.
All right, awesome. Let's see. Oh, this is kind of cool. Fifth graders, they would like to know, do you retrieve the tags after you put them in the fish?
Another good question. Yeah, so what we do is when we put the tags in the fish, we also put an external tag. It's called a Floyd tag. It's like a little brightly colored tag that we put just above their back by their dorsal fin. And that allows us to understand whether or not we have an internal tag in them for tagging purposes. We don't always get the tags back because we don't always retrieve the fish. But when we do, we definitely retrieve the fish and we'll remove the tag from the fish. If we don't remove the tag from the fish because we don't recapture them, it doesn't hurt the fish, just kind of becomes a part of their body over time, and they're typically fine by it. There are species, which we don't tag very often, that actually we put the tag inside of them and they actually expel the tag or they spit it out of them through natural processes. Those fish that we tag, we actually use a slightly different technique for tagging so that the tag remains in the fish.
Okay. So this is, I'm just thinking about how you tag the fish and what you're using. It's not like a satellite tag. You've got receivers. So if the fish is in the area, they ping?
Yes. Yeah. If a fish was within the range of the receiver, the tag will go off at, you know, predetermined intervals, you know, a minute or so. If the fish comes near the receiver and within range, it will ping, and then we get the number of the fish and know that they're there and then for how long.
All right. Because I was going to ask, we've hosted some shark researchers using satellite tags, and they've gotten really weird readings where, you know, the sharks have been caught and travel over land and things like that. But they'd be too far from the receiver, so you probably don't get any strange readings if your fish are caught.
Yeah, we also do have what's called satellite pop-up tags that are attached to the fish, and then at some point in time the tag releases, goes up to the surface, and then we can see where the fish has been over time. So a little bit more similar to what you're talking about maybe with sharks.
Okay. That second grade class would like to know about what you're eating in the field.
That's a good question. What we're eating in the field. Even in really remote areas, we eat pretty normal food. We just have to bring it in by helicopter and try to keep things a little simpler. But we usually do have cooking facilities, so we try to keep meals simple, like spaghetti and meat sauce, or spaghetti and tomato sauce. Things that are fairly simple to cook. We're not cooking cakes or anything in the field like you would do at home, because it's a little bit more difficult and we don't have time for that and don't always have the facilities. And then, we do have goodies and such, because we're burning lots of calories when we're in the field. So, I like peanut M&Ms, and then we have beef jerky and sandwiches and stuff like that. So that's kind of what we have while we're having lunch in the field.
All right, let me go back to our YouTube chat. We talked about the packs. Okay. So this is a curiosity question. What's the best or most exciting fish you've caught? Maybe just on your own time.
On my own time. I mean, the fish that I catch, hopefully you got it from the videos. They're pretty cool, especially that second video. One of the coolest fish I've ever caught, and it's always really cool to catch them using our backpack electrofisher because it's really works really well. And so if you like fishing, you kind of look at a spot and say, I think there's going to be fish in there. And then you basically get to go catch them. But on my own time, fished in tropical waters and caught some tuna and trevally and stuff like that. And that was pretty neat too. So, you know, that was near Hawaii. So yeah, it's kind of cool. But I've never caught some like enormous fish, like really, really big, which is sort of something I'd like to do someday.
All right. I've got one more question here in the here in the chat, and it's about the pucks that we saw in the video. Do you have any results yet from those experiments?
Yeah, we do. So the master student that was leading that project, they actually just went out a couple of weeks ago and they dug up those pucks, and they found that survival was quite high, around 75%. And so that's fantastic. It tells us that we can take eggs and plant them elsewhere in rivers for this species so that, you know, we can successfully have eggs incubate and then juveniles emerge and then contribute to the population. So we're really excited about the results. They haven't been published yet, written in a report, but really promising results so far. So it's very exciting for us with that study.
All right, Neil, I'm going to ask you one more question. This is one that we always like to ask people who join us. What inspired or led you to a career, studying freshwater fish?
I always loved the outdoors, and I didn't fish a lot when I was a kid. Went a few times with my uncle, and so then I started fishing more. And I just kind of thought, yeah, it'd be really cool to have a job where I could catch fish and study fish. And you know, it inspired me to go forward and pursue a career in this, and kind of went from there. So I really feel fortunate to have a great job. It's a lot of fun going in the field. I get to go to cool places. So that's my story.
All right. So we're going to start off with a shout out to our classrooms. Thank you so much for joining us today. We hope to see more of your classrooms as our Canadian Environment Week continues, looking at forests and freshwater and ocean and the Arctic and our air, all 5 things that we have in abundance here in Canada. So Neil, as always, great to steal a little bit of your time. Thank you so much for sharing some of the exciting freshwater fish work that you're doing in remote areas. And thanks for doing some of the Q&A with us today.
Yeah, you're welcome. It was a pleasure. Thanks for having me.
All right, we're going to sign out for now, but I'm sure we'll see many classrooms as the week goes on. Thanks, everyone.